Hi, everybody. My name is Steve. I'm here from Cyclica. Cyclica is a structure-based and AI-augmented platform for drug discovery, and I'm going to talk to you today about empowering on-demand scientific computing with Google Genomics Pipeline. So, a little bit more about the company. We've been operational since 2014. I joined around 2 years ago, personally. Our technology has all been developed and patented in-house. We've been validated and commercialized since 2015, and we're working with some of the top pharma and academic and biotech groups in the world, which is very exciting. There's 31 employees, computational scientists, experimental scientists, data scientists, software developers, and we've raised a total of $12.5 million to date. I'm coming to you from Cyclica's core technology team. We're responsible for sort of the most technical aspects and least scientific aspects of what goes on at Cyclica. As I mentioned, that's me. I'm Steve Constable. I'm a computational biochemist by training, so a mix of computer science and biochemistry. I'm the team lead, and I have a love of making things work and getting things done. Simon is a very talented full-stack developer, and he's actually written most of the code you're going to see today. Jonathan is our DevOps guru. Like, we'd be nowhere without him. He does all our Ansible, makes sure all our servers stay on, makes sure all the deploys go great. Love having him around. And Cindy is our newest member, actually newest person at the company. She's a junior developer right now. She is mostly learning the ropes of what we do and what we've done so far, and she's aiming towards front-end development. So, let's talk a little bit more about the problems that we're trying to solve. This is a common patient story. Many of you may be familiar with it. So, we have Alexis. She's diagnosed with a heart condition. Very sad. She's prescribed a pill. She takes the pill as prescribed by her doctor, which is good. It enters her bloodstream, goes throughout her body. She starts to feel great as the medication has its intended effect and improves her condition. Unfortunately, as a side effect, she begins to have severe abdominal pain. We don't like side effects. So how can this have been prevented? Well, to understand that, you have to understand a little bit of biochemistry. So for those who are new to the idea, this is going to be your crash course. So, pills are full of small molecule drugs. When you take a pill, they are released into your body. This is how we represent a single drug molecule. It's very small in real life. What drug molecules do is they interact with proteins in your body. Your cells are full of proteins. And when people think of protein, they think of muscles, but that's not necessarily true. And I was challenged by one of my colleagues at Cyclica, Steve, you're giving this talk to a bunch of developers. How are you going to explain to them what this slide really means? And so here's my analogy. So proteins are little EXEs. They're executable programs that the cell makes. The source code is your genes, your DNA. And the compiler is the ribosome, believe it or not. So when we introduce a small molecule, what we're really doing is feeding some input data with the hopes that this particular input data will trigger a bug in the EXE and cause it to crash. We call that inhibiting the protein. Why would we ever want to inhibit a protein? Well, if you have a genetic disease, that means your source code has become corrupt and the EXEs that are generated are not behaving properly. So we obviously want to stop those. Furthermore, many of the cells that the drug would come in contact with may not be your cells. They may be bacterial cells, in which case we definitely want to stop their proteins because we don't want them to be alive. That's how antibiotics work. So throughout the human body, we have various tissues, each of which is composed of different kinds of cells, which are full of different kinds of proteins. We design a drug to target a specific protein, but often it has other side effects that we don't want. This is what's responsible, the physical basis for side effects in medication. A small molecule will likely interact from anywhere between 30 and 300 separate proteins within the body. In addition to these problems, the cost, if you're likely aware, to bring a new drug to market is very high. We're talking about 12 years of work. We're talking over $2.5 billion in research and development. And in addition to that, we've seen an 80% decline in productivity since, I guess, the peak of drug discovery. So at Cyclica, we're trying to remove the mystery and get drugs to the market faster and at a lower cost. This is, I'm sure you Googlers will be aware of the phrase moonshot. So our moonshot is to bring the time of drug discovery specifically, not the testing and all of that, down from 7 years to 2 years. That would be great. We would love that. And our Mars shot, which is even beyond that, is to design drugs for patients, not just proteins. By that I mean by accounting for all the different proteins within the body, not just the single one you're targeting, and beyond. So here's some excellent history on computational drug design. As you can see, way back in the 1980s, people were working on this. Things have come a long way since then, but this has been a very well-researched area. You can see back there, there's the simplest, drug interacting with a single protein model that I could even ever imagine. That was before I was born. So at Cyclica, we believe that AI is not a silver bullet. We're not going to be able to replace pharmaceutical scientists. We need a ton of data to make good predictions. We want novel predictions, not something that's already known. We want to use AI to augment biophysical and chemical informatics approaches, such as in our flagship platform, Ligand Express, which I will introduce now. So Ligand Express is sort of our first tool that we brought to market. It's a science and user-facing web application. Scientists can upload candidate drug molecules, and Ligand Express allows you to investigate what kinds of proteins within the body might be interacting with this particular drug of choice. You can actually use this today if you're a scientist and you have a deal with us. So this is what it looks like, sort of your first login screen. This is basically our administrative dashboard. You get to see, what kinds of experiments have been run. You have different projects, which are like initiatives that you're working on. You can manage your users, et cetera, et cetera. We also, our newest addition down there at the bottom right is property prediction, such as the likelihood of a molecule to cross the blood-brain barrier. It's an important property. It's not really governed by interaction with a specific protein, but we still need to know about it. And when you really look into what Ligand Express is providing you, this is what you get. This is what we call our analysis view. So on the left, we have a ranked list of every protein in the human body, ranked by how strongly we believe that your target molecule interacts with that protein. In the center, we have a fully 3D structure visualization. The protein that we're analyzing is shown there with where we believe the drug is interacting with it physically. Protein is in red. The drug is in gray and blue for carbon and nitrogen. And the white highlighted area are the specific parts of the protein we believe are in direct contact with the drug. Furthermore, on the right-hand side, you can see some information about this molecule that we're analyzing. In this case, it's LSD. Below that, you can see a summary of the protein that we believe it's interacting with, which in this case is, I'm not expecting to remember this, the 5-hydroxytryptamine receptor 2B. And at the bottom right, you can see our effect prediction. This is an AI technology that analyzes many known effects of drugs with proteins, and it has decided that LSD is a molecule that is likely to modulate the effect of this protein, so change it in some way. And the 4 molecules shown are the 4 closest molecules, and that's why we believe since LSD is so similar to those known interactions, that's why we believe that it will have this effect. So we're not just telling them it will, we're showing the scientists like, hey, these are the molecules we think are representative of this interaction, and it's up to the scientists to say whether or not they really believe that. So in addition to that, we can go into our network analysis view. So here, we've taken that list and we've filtered. We're using our expert domain knowledge that LSD can be associated with schizophrenia, bipolar disorder, depression. And so given that, we're showing the related proteins to those diseases that we believe are interacting with this LSD molecule. You can see the connectivity here. And each of these links is not just proposed by us. You can actually click in and it's backed by scientific literature. So we can put the power in the hands of the scientists to decide whether or not they believe that link is true or not, which is very important. We're empowering scientists, not replacing them. And this is our newest addition to Ligand Express. So I mentioned earlier about the blood-brain barrier. This is LSD again. And hey, look, we believe that it's going to cross the blood-brain barrier, which is good because we have some pretty good evidence that it does. And we have the most similar molecule that we know crosses the blood-brain barrier, according to this model, and the most similar molecule that we know does not. And having these structures allows the scientists to decide, yeah, that looks right, So Ligand Express has been on the market for years, and we've done a lot of work on improving it and trying to get it to be the best it can be, and we've learned a lot of lessons along the way. Which brings us to our next major product, which is called Ligand Design. And so for Ligand Design, we're taking those lessons we've learned and we are applying them to design new drug molecules essentially from scratch. And so the reason why we want to do this is we want to be able to suggest new drugs that we think will be effective and also safe. So avoiding proteins that we don't want to interact with and definitely interacting with the ones that we do want them to interact with. So I'll talk a little bit more about how this works. So Ligand Design is a multi-generational process. A single generation has the following inputs. We start with a seed molecule, which for best results is something related to the area that you're studying. We start with a list of targets, which are proteins that we do want to interact with. These would be those that are related to the disease that you're trying to treat. And we have a set of anti-targets, which are proteins maybe that we know are related to the existing side effects of existing drugs on the market, and so we want to avoid those. The algorithm will take the seed molecule, or more than one, fragment it, recombine it, filter the resulting molecules, predict their non-specific properties such as blood-brain barrier crossing, and finally analyze which proteins they think the drug will interact with. So to rehash that, fragment the molecules, filter the molecules, select favorable properties and toxicity profiles, analyze whole body protein interactions to come up with a new set of molecules, and then this process repeats and it repeats and it repeats and it repeats. And at the very end, what we deliver to the client is a list of about 200 molecules that are like, hey, these are some molecules that we think are going to be really good for the specific thing you're interested in, and now it's up to you to use your domain knowledge to be like, what do you think about them? Pick the ones you like the best and maybe use those as a jumping point for something more. So the number of possible small molecule drugs is very large. There's about 10 to the 60 of them. Not quite a Google, but it's a lot. The The space that we think we can search within ligand design is about 10 to the 30, the hypothetical space. The number of molecules that we'll process during an actual real run is around 4 million. And the output, like I said, is around 200. And we do this in 20 hours of compute time. So we're going to change gears a little bit. So I mentioned earlier that I'm in the core technology team. I personally don't interact with a lot of the scientific-facing code, but all of this code needs to get executed. And that's where my team comes in. And I'm going to talk specifically about Newt, which is our new high-performance computing system that we're working on. So for those of you who are not familiar with the ins and outs of distributed computing, what is it? Why would you want to do it? So many of you, I'm sure, have used Google Compute Platform. You can select what kind of machine you want your VM to be. If you look on the little selector, you can probably get up to 96 cores. Or maybe if you're doing a custom machine type, you can get like 140, maybe a little bit more. But eventually, you will reach a hypothetical limit where your virtual machine has used up all of the CPUs in the physical host. So how can you go beyond that? The idea is that by distributing tasks amongst a network of machines, we can in principle use arbitrarily large amounts of CPU and disk. And what makes this a great approach in our field is that many of the tasks are what you call embarrassingly parallel. The individual computations don't depend on each other at all. And so the realized speed up scales linearly with increasing numbers of CPU. So the system for Ligand Express that we had and continue to use today, that we're moving away from, looks something like this. So a user logs into the web server and submits a molecule for execution. That web server uploads some input files to Google Cloud Storage. From there, the web server additionally requests the creation of a new VM template, the creation of a head node VM, and creation of an instance group for workers based on the VM templates. So Google magically delivers those to us. We get our head node, we get our worker machines. Those start up and begin processing the input files that we uploaded. There's a lot of data transfer. This is where the magic happens on these VMs. They run all of our scientific code. And in the end, they write the results back to Google Cloud Storage. Finally, the head node VM takes those output files, reads them, and injects them into the web app via a Django REST API. And those results get delivered to the user. So some issues we've had with this setup. Number one is if resource overutilization occurs, i.e. too many jobs are submitted at the same time, then everything in the system breaks. Because the new VMs, they fail to start up, and then maybe some of them start up and some of them don't, and you end up in this weird inconsistent state. And then I have to jump in and start deleting stuff and restarting it. Side note, being able to dynamically resize instance groups has been a lifesaver for me. I love it. So one of the features that I do love. Also, the images that these VMs are based on can be kind of a pain to manage. There's, from what I can tell, the best way to, if there's a dependency update, for example, to update the image is to start a new VM based on that image, install your updates, shut it down, and then mint a new image. So it's kind of like this living entity. And occasionally we've had issues on Ubuntu 18.04 with downloading data from Google Cloud Storage on startup. It's not fun, and that has caused us some headaches as well. So why do we want to move away? The code that runs on those VMs is basically a monolithic application. It's very specific to solving this problem. As I said, we have Ligand design now. We need to think about generalizability. We have hard-coded compute API calls. We have legacy VM images. We have new internal applications being built that are run on demand rather than all at once when you upload a compound. So we want people to be able to pick and choose what kinds of properties are computed rather than doing everything. And in addition, thinking about investment in the future for our company, we want to be able to facilitate rapid development of future apps that require these high-performance computing resources. And this is literally the original design description I got from my old manager of what we would be building. And this is the workflow. User logs in, you can view the queue. Here's the kinds of properties that would be viewable. You could view existing pipeline scripts. You could create a new one. You could start a pipeline with some Google Cloud folder for input and some other Google Cloud folder for output. You'd view the queue. Probably you only want to see things that you're interested in. You would spam F5 to refresh and wait for it to be done. Delete and queue jobs that you're no longer interested in, and finally get an email when the job is done. So we had a great plan for this. We were going to use this great software we found on the internet called Toil. It's an HPC workflow library. We actually named our system Newt in reference to Toil. It's a Macbeth joke. I don't know how many of you are familiar with Shakespeare. So Toil advertised they had cross-cloud compatibilities. They had this sort of cloud and system agnostic way for running cross-platform workflows, which is called Common Workflow Language. It's very popular in the industry. And it seemed to be simple enough for our usage. There are some competitors in this area you may be familiar with, Airflow, Luigi. They were ruled out because they were too complicated for us. Unfortunately, when we went to actually implement things, we couldn't get Toil to work at all. We had problems executing the required version of CoreOS in our Google platform account, which is very sad, but the day was not lost. I personally had an interest in this open source project called dsub. It is an homage to the famous qsub command, which I'm sure all of you know from when you were in grad school in biochemistry. It is very popularly used to schedule jobs on Linux compute clusters. So dsub is itself a job submission application. It uses the Google Genomics Pipeline on the back end. The important thing is we tested it and it worked, and I was very happy. In addition, one of the critical things we're looking for is that within a submitted job, you can then go back and submit a sub-job, which allows us to dynamically spin up a whole fleet of VMs without prior knowledge on exactly how much would be required. So this is the Google Genomics API. One of the interesting things that you'll note is it's not exactly tied to genomics specifically. This is a general purpose batch computation framework that anyone can use. We actually initially shied away from it internally because it said genomics, and we're like, well, that's not really what we do. But after going through it, I was like, no, this makes a lot of sense. So you can run a pipeline, you can cancel a running pipeline, you can check in and get the status of one that's running, you can list everything that's running, and we finally have a method that allows periodic status updates from the VM. And dsub is a wonderful application. It's just a simple CLI. It drives this API. It supports job submission, job status queries, and canceling. All jobs run inside a Docker container, and all input and output files are read to and written from Google Cloud. It supports many of your familiar features from GCP. We have all the same machine types. We can do attached disks. We can do GPUs. The networking is similar. You can set up preemptible VMs, which allows you to save money. And it adds the following features on top: automatic handling of I/O, automatic startup and shutdown, and automatic retrying of failed jobs, which is very important. So what we did internally is we started with this application, and we go, okay, well, we don't really want a CLI. So we're going to rip off that CLI layer. We don't want that anymore. And we're going to replace it with sort of a Pythonic library interface. So for example, this little snippet of code which submits hello.sh for execution goes from a line in bash to a Python library call using Quargs. So we're gaining all the functionality that we need. And in addition, we have this constraint that all the data must be loaded from GCS. So this is all functionality we essentially got for free, which is great. So as you can see, we can submit a job. We're provide some metadata information. The provider is sort of where it gets run. You can do Google or local. There used to be multiple versions of Google, but now there's only one. User, the project is like your GCP project that you're working in. The Docker image can come from Docker Hub, but since a lot of our code is proprietary, we want to keep it private, we're actually using GCR, which is the Google Container Repository, and it works great together. We set some environment variables that we need for our code, set up output, Google Cloud, or Google Cloud Storage folder for logging. We set the zone that we want to execute in, and then we tell, we pointed out where we want the input files to come from, where we want the output to go. Our command, which is what actually gets run, that's the important thing. The machine type to use, so that's how much CPU, RAM we're throwing at it, and the size of the boot disk to use. So we got all that for free. We're very happy about that. But we need something to actually run, right? We need a script. So what kind of script are we going to use? We made a pipelines class. So unless you have a script, your spawn job isn't going to do anything. It's just going to terminate. The sort of lingua franca is to use bash or in a bash script to do your work. But all of our devs are Python devs. They want to use Python. Fine. So we provided a pipeline class that tries to automate as much work as possible for argument parsing and whatnot. And the scripts that we run on Newt are all subclasses of this pipeline. And here's the code example. This is it. I hit a lot of the boring implementation details, but there's basically a couple helper functions, some internal variables, and then when you want to subclass this thing to actually do something, you fill in the bottom three methods: parse data, parse download, and run. So a real live example of something we actually use looks like this. So this is a pipeline that performs deconvolution, which is a very technical thing we don't have to get into. But as you can see, we have a constructor. We read in some parameters, set some internal variables, handle downloading some dependency data. And then finally, the magic is just calling some in-house Python code at the very bottom. Now, because a pipeline script is like an application, we want there to be free parameters. Like you wouldn't want a command line app that just does one thing and can't accept any input. So in order to supply parameters, quote unquote, at runtime, we decided to use YAML files. We invented this parameters.yaml. It's logically equivalent to command line arguments. We basically upload this to the correct location within GCS, and then when the pipeline runs, it runs with those parameters. It's a simple list of key value pairs that provide input. An example is like this. So this is for deconvolution. And so if anybody within our company wants to run a deconvolution platform with high performance computing, this is it. That's all they got to do. Upload this and run it. It's great. This is our beautiful web UI for the Django app that's responsible for driving this. The fields are pretty simple: user, name of the pipeline, the location in GCS of the script to run, which is a pipeline script, the machine type to use, the Docker image to use, and a version field for tracking that. And then when you go to run, so this is like, we call it a pipeline job, which is like when it's actually running. You need the pipeline to run, your username, the name of the project, which is where we store the input files, and we get a status, a priority, and this is sort of the job ID from within dsub that we hang on to manipulate the running process. In addition to all of that, we of course, we don't want everybody who needs to do anything to have to log into a website to get things done, because we want this to be automated, right? So we have a command line app, and we have a REST API. This allows you to submit jobs from within jobs without needing to use a website, which is very important. So this is the command line app. It's very simple. Little things: auth, cancel job, list of Docker images, download output from a job, enqueue a new job. That's the big one. And the rest is all basically managing that. So, of course, the big selling feature for us is that jobs can submit their own jobs. And this is literally the code. This is all it is to do that. As you can see, we have a new job function, gets an environment variable, uploads a parameters file to Google Cloud, uploads input files to Google Cloud, and then enqueues a subjob. It's magic. So to recap the work that we've done, we've re-implemented everything that we had in the original legacy pipeline. Each individual step itself is quote unquote loosely coupled via this enqueue function in a way that allows us to call each component separately. And so what else can we do? We can do on-demand molecular property calculations. We're doing hyperparameter optimization of neural nets, accelerating that lag and design process that I mentioned earlier, obviously doing deconvolution, as I showed in my examples, and there's more coming to be sure. The advantages of this, we only need one server to run 24/7. All the other machines are only started when they're needed, and this allows us to maintain our low overhead cost structure that we love dearly. In addition, the sort of built-in queue in the Genomics Pipeline API provides like this safety net that stops us from accidentally trying to use too much resources and getting into an inconsistent state. The pipeline-derived code, it's very easily reusable for us. The average turnaround time for a new feature request is a day. So it's easy for our devs to get new things up and running. And finally, I'm very happy that instead of using these Google VM images, we're now using Docker images, and we have a Docker file that's saved and gets reused. It makes it easy to reason about the different images that we have. There are disadvantages, unfortunately. So I'm not in love with the idea of passing parameters around as YAML files. It's not a great solution, but it does work. I'd like to improve this to the point where you can literally just call the enqueue function with the name of a pipeline and your arguments in Python, and everything else will be handled for you. In addition, the web UI, it's not great. It doesn't have a great user experience. It's mostly used for internal testing. We want to have some design work done to make something that's truly usable. We had the idea of having some internal staff use this to get some work done who were not computational people, and I shot that down immediately. I was not going to have that happen. And also, it's not easy to dynamically resize the number of VMs anymore, as we could using instance groups, but we can cancel subjobs, which is nice. Things we want to get going. So, in the future, priority queuing, it's important to us. If we have a user-facing job that would take nominally 10 minutes, but it's in queue behind a hyperparameter reoptimization that's taking 5 hours, we obviously want to bump up the user-facing one first. So we're building our own priority queuing system that's basically going to act as a double queue. The priority queue will enqueue things in the proper order inside the genomics pipeline. We mentioned earlier we like the idea of supporting this common workflow language for interoperability between different sort of compute resources, so we would like to re-implement that. There's also the irony that we're building a pipeline system on top of a job submission system, which is built on top of a pipeline system. So we could remove dsub as sort of a intermediate layer and rework it so we're making direct REST calls to the Genomics Pipeline API, but that will be done in the future. We could also make more effective use of network addresses. We don't need a network address every time. And I'd like to do more experimenting with preemptible VMs to save money, and this retry option, which promises to automatically retry if your machine is preempted. Messages to Google. So take home from us, thank you very much for this system. It's wonderful. We love the Genomics API. Please keep working on it. We think that it could be more powerful if it supported priorities out of the box. Then we wouldn't have to implement a queue on top. And finally, if the dsub library itself had better separation between the CLI and the actual functional code, then we wouldn't have to hack together a library from it. We could just call the functionality natively. So it'd be nice to see that too. Finally, I want to thank the Google Genomics team, the people who contributed to dsub. I want to thank all my colleagues at Cyclica, And I want to thank you all for inviting me to talk.